see better animations, which will hopefully enhance your understanding. But first, I want to finish effectively the passive motion component. And so we let enough air into diffusion. I don't think you have to worry too much more about that. But from that point, we're going to get on with facilitated diffusion and osmosis. And those are the other two passive movements of molecules. And it starts to get really important, everybody, as we're getting here now. The material is going to start to build on itself and become more and more complex as time goes on. Thank you. It's okay. It's okay. Moving on. So, just begin. Effectively, they have already have another one of these many PowerPoint errors. You can see it right here. Hydrophobic molecules. They're not. When we're talking about facilitated diffusion, we're talking about hydrophilic molecules, of which glucose, amino acids, and ions are all a subset of. So, starting with that, effectively, facilitated diffusion involves some kind of a protein passageways, and if it implies substances that change with protein carriers, this is something that effectively indicates that it's called carrier-mediated facilitated diffusion. Not a big deal for us. Or channel. And to me, channels basically imply that it's always open. This is not a distinction I would ask you about, but you will see that we'll start to see the nuances of these as we get into the applications. This is more definitional stuff. And to me, those definitions are a little bit confusing, to say the least. So they're effectively, all of these are integral proteins. Those integral proteins allow materials to pass effectively through them, with or without changing shape. If we're looking at something like a pore, or what they call an aquaporin, that's going to maintain its shape for water. And nearly all would be a fair way to express it. Cells have that capability. When we're talking about those junctional proteins, like desmosomes and tight junctions have nothing to do about water getting through the cell. The junctions are between the cells. So effectively, the water can squeeze in between the cells in the absence of sufficient amount of tight junctions, which, and frankly, that's more often the case than not. But these are very, very specific, these carriers. They're all about these polar molecules. A lot of them are sugars and amino acids. Basically, it's a size type of situation that's involved with these as well. And every so often you reach a point of what's called saturation. There's only so many places, but you can see that's really none of this is really a big deal in what we're studying now. It's just more traditional anatomy and definitional things. You're looking at something with a shape change there. You can see obviously it looks like a sugar because it's hexagonal, and they're just trying to demonstrate, even though that's not really how it happens, how the stuff gets through. Okay, so the channels, again, their water is effectively the core. They're aqueous materials by their very nature. Depending on the size, charge, and variables, as you're going to see very, very shortly in an animation I'm going to show you, this is all about how sodium and potassium create something called the resting membrane potential. And I mean, it's the major building block. We start beginning to look at nerve and muscles. The whole second half of our course is based on variations in what's called resting membrane potential. And you're going to get sick and tired, as I said before. When we're looking at these waters, sometimes we call them leakage channels, because things can leak in and things can leak out. It's bidirectional. And you're going to see very shortly it's about what kind of channels they are and how many. It's just something that can leak out of the cell or leak into the cell. And it can certainly go in both directions at the same time. And then down the road, when we start talking about nerves and muscles, you're going to be, again, sick and tired of hearing about gated channels, chemical or ligand gated channels, voltage gated channels, which have to do with electrical signals. That's how things work, particularly the major systems about nerves and muscles and signaling. It all starts with what's the cell is like. Not every cell can do this. Nerve and muscle cells have a membrane that can rapidly change. And because it can rapidly change and return to its resting site, it has the ability of sending that signal along its membrane, and in doing so from cell to cell, or from cell to an end organ or an end object. It's like having a still pond of water. You just throw a pebble in there and you watch the ripples. It's not unlike that. Waveforms, the way electrical energy frequently moves along. That's more physics than chemistry. And I just want to give you an analogy. So here, typical channel. Okay? If it's open, it'll get through. That's the whole idea. Facilitated means there's something in there that facilitates or enables it. This is different. So far, with diffusion, simple diffusion, and facilitated diffusion, we've been talking about solute, dissolve, universal form. So all of this is solute. Those are the terms I use when we get to not this example, the next one. Solvent. That's the more appropriate term to understand it. So the solvent is water, and the solute is what's dissolved. And when we talk about osmosis, we are basically looking at the concentration of the solvent. When you have, imagine you have two identical vessels, containers. Let's say they're 1 unit, 1 liter, 1 gallon. And you're going to put, they're not chained together. You need to ignore those little dashes. You can put 9 things in one and 3 in the other. So they're solutes. They could be sodiums, they could be chlorides, they could be potassiums, they could be sugars. Okay, when we start to look at solutes, understand we're looking at the number of solutes when we talk about osmosis. So down the road, you're going to see, and it's really not relevant now, something called 300 millismoles. That's the number we give in our bodies, inside the cell, outside the cell, for the number of solutes dissolved in X amount of water. It's a consistent number that's really important. That includes anything. So when salt is in there, and we have a lot of salt, it's not one salt particle. When it gets into water, it forms 2, sodium and chloride. When it's glucose in water, it forms just 1, a glucose. So depending on the chemical construction, what happens is it stays, when it dissolves in water, this stays 1, or if it's calcium chloride, now it becomes 3, calcium and 2 chloride. You get the idea? So that's when you go through all the chemistry and things like that. But effectively, that's sort of what I'm driving at here. So back to this example of water moving. You can see here, just by sheer numbers, I got 9 things in here. I got 1 vessel, 9 things in 1 vessel, versus 3 things in 1 vessel. I got a lot more things here. I have a lot more solute proportionally. Here I have a lot less solute. But what is implicit and obvious, I got a lot more water. Just flip it. Here you can flip, you have 1 unit of water with 9 things in it, 1 unit of water with 3 things in it. I know where you come from. Where I come from, 1/3 is always bigger than 1/9. That's just how it works. So proportionally, you have more water. If you had a magic doorway, and these things could expand, the water would rush in to balance it out. Living systems are like that. We've been talking about diffusion and facilitated diffusion, higher concentration, lower concentration. That's solutes. Water behaves the same way. Dynamically, water is moving, balanced concentration. Because of this, okay, we're going to see this kind of comes down to something called tonicity. Tonicity is this. It's the fact that whenever we want to put fluid into a system, okay, inevitably, when we start intravenous, we start with something that's called isotonic. The classic examples of that, we call it, and it's the reason why we call it normal saline. Normal saline is 0.9% sodium chloride. A tiny pinch of salt, 0.9 grams, very little amount of salt in 100 milliliters of water. Years ago, we used to have what's called D5. This is called normal saline. The normality has nothing to do whether it's normal salt or special salt. It means that normal saline has that. You process it by the math and the number of particles, it has 300 million dollars. Years ago, we used to do a lot of sugar water. It's called D5W. Dextrose, the active part of glucose, 5% in water. What you think you see a lot with burn patients and really rapid fluid loss is called lactated ringers. That's a solution. It's too complicated to put down. I certainly don't remember off the top of my head. But it's got mixtures of salts and sugars and acids and bases to try to mimic what your plasma is like. Yet, same osmolarity. And so the whole idea is if you're using something like that, they're isotonic, they're either going to push fluid into the cell or pull fluid out. If you put too much fluid into the cell's going to burst. That's the difference between our cells and animal cells and plant cells. What's the biggest difference? Everybody learns this in biology. Plants have a cell wall, right? We have a deck full. And the plants, my wife is right on top of watering them. She's very, very, we're growing stuff out there. Okay? Our peppers are looking good. So nevertheless, what happens when a plant loses water? It droops. You can tell it's drying out. So what do? We water it. Okay. What does the plant do? The water gets in there to the cell because of the cell wall. It pushes on the cell wall. The plant, happy. I'm happy for it. I just got water. I'm ecstatic. Go ahead, water me. Make my day. That's what a plant does. For us, you can overhydrate. If you don't believe me, here's a class. This actually happened, maybe 20 years ago. I'm trying to get this. I'm going to tell you between 15 and 20 years ago. It was when the Wii gaming system came out, WII. Okay, some disc jockey in the LA area thought it'd be a clever idea to do an on-radio promotion. I'm going to have 2 people come on, and they're going to drink as much water as they each can. Whoever drinks the most water, we're going to give them a Wii system. They drink the most water without peeing. Hence the name Wii, the Wii system. The lady won the system for her kids. Good news. Bad news, she died about a day later. So don't think that this is something innocent. It can kill somebody. Overhydration as much as dehydration can kill somebody. And so that's where you get into the ideas. What I was showing you there are called isotonic. The tonicity, they're like imagine the cell having muscle tones. Probably where they derive. At least they would make that allegation. I don't know if that's necessarily true. What plain water was that this lady was drinking is called hypotonic. It means it has less, the solute number or less proportionally. So it's more likely to push water into the cell, and those cells, boom, explodes. Seawater, hypertonic. It's going to, because of that, the water is going to be pulled out of the cell. The cell will shrink and also be destructive. That's why we're so worried about dehydration, particularly in people who can't compensate, the very young and the very old. Those are different kinds of media. That's why we use isotonic media. That's why this osmosis stuff is so important. Blah, 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 aquaporins. Here, they'll show it to you. So we measure it in this way. Varies with the number of solute particles because they displace water. Okay, all good. But this is basically the crux of what you have to know. That's why I try to simplify osmosis. It's the movement of solvent. And when, not this exam, but the next exam, that's the way it will be expressed. The movement of solvent or the movement of solutes. So you need to be familiar with the terminology, at least for me. Okay? The clinical application is extraordinarily important because you're going to be treating people. That's why when we start an IV, unless otherwise specified, you've used isotonic media. And very rarely, the way you vary what's going to happen in the cell is you add stuff. Occasionally, we used to use ringers and half normal saline when I was a resident. That's almost 50 years ago. I'm sure these things have changed. So my knowledge of current medicine is unfortunately as a receiver rather than a giver, if you know what I mean. I don't like that part nearly as much. So everything seeks equilibrium. So if you had a magic membrane, and obviously you have more particles here, okay, what happens if you flip the switch and open the membrane? Everything balances out. Equilibrium means it's still moving. If it stopped moving, the person would be dead. So in living systems, a particle is moving in one direction, water molecules are moving in the other direction, and they keep doing that, but the net movement is zero. That's what's meant by equilibrium. On the other hand, okay, different. So water does the same thing. If you had that same magic membrane in the same illustration, and you open that membrane, you flip the switch, the water would move to balance it out because the particles are too big. So the water moves, and look what happens. It'll change the volume. That's why I'm going through that whole business with the cell exploding. And you can see it here. Water has a thing called hydrostatic pressure. So your blood pressure is hydrostatic pressure. When blood goes through a blood vessel, here's a blood vessel, like this. It doesn't just go linearly. It pushes against the walls. You all know how blood pressure is taken. You put a cuff around the appropriate part, and you inflate it. And then as you gradually release the pressure, when it reaches something we call systolic, the fluid pressure is able to push that open, and you'll hear a sound. That's the start. We call that the systolic sound. When you stop hearing the sound, you get to the bottom of it. Because then effectively you release the pressure so much that it can't create a force and hear a quote pulse or sound. So that hydrostatic pressure is always, normally when you get to a capillary, it's got a ton of pores and a ton of openings. Water will seep out like crazy, but we have stuff in there holding the water, mostly proteins, but all of those other solutes act, and we call it an osmotic pressure. It holds on to the fluid, and it's a tug of war when you get to the circulatory system. And why this is so important, when you get to capillaries and circulation, which you don't do until AP2, now you would be able to understand at the start of a capillary bed, which is arterial blood, the forces are overcome, and at that point there's enough of a pressure differential that it'll push fluid out. But at the end, having lost that fluid and the nutrients and the oxygen, blah, 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 it's going to start pulling back in at the end of that capillary bed, and now it'll pick up waste products, additional fluid to restore the fluid balance, and then at the same time, carbon dioxide will come along for the ride. That's the beauty of what a typical capillary bed, you're doing it right now, because your systems are functioning, even though you're not necessarily physically active or very physically active, so to speak. So those are things, and you're going to constantly be familiar with. That's why I want to give you an idea as though these are really vital building blocks. So water equals, that's plants only fill so much, so the water will stop. Plants have these interesting thing called vacuoles, which we don't have in our cells, that actually pump it out. That's why the plant doesn't explode. You could overwater a plant, but I think it's more of has an impact differently, and I don't understand plants that well to be able to tell you that. That's why I have a black thumb. Unlike a green thumb, don't give me a plant. It's gone. So if my wife has to go away, she has a very, just such a romantic way of telling me this. I will kill you if you kill my plants. You probably have heard that. We dads are notorious for that. Animal cells do not have cell walls, and blah, 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 blah. So here are the isotonic, hypertonic, hypotonic, okay? And what happens to the cell, in the fancy words, when it shrinks, it's called crenation. You'll see that in a second. When it bursts, because the plasma membrane bursts, we use the term plasmolysis. You can pronounce it plasmolysis if you want. It doesn't matter to me. Lysis means bursting. Plasma implies the plasma, which is equivalent to the cell membrane. Again, those are terms that are used interchangeably. Where am I? Here we are. And this is the whole idea of osmolarity, number of particles. I do this experiment. And when I do my advanced phys, we get aseptic blood, and I have, test tubes with just plain water, deionized water, okay, so it's hypotonic, normal saline, and I have extra salt, so making it more like seawater, 3 to 5% rather than 0.9. If you put it in isotonic, the red cells don't change. We look under a regular microscope like you would do in the laboratory. It looks exactly unchanged. When you look at it, now these are micrographs. So in the micrograph, you don't see this kind of definition. If you see it in micrograph, you see it under a regular light microscope, under high power. What you see when you use it in salty water, you don't see those protrusions, but you start to see the roundness disappear and it starts to square off. That's how crenation looks two-dimensionally, if you want to think of it that way. And you can do this. I am super fast. I'll flatter myself at focusing a microscope. I am really quick at that. I've been doing it for a very long time. I can't even see it enlarge. By the time I focus it in a matter of seconds, after putting it in pure water, it bursts. So all you see is nothing. All the red blood cells are gone. I mean, I've had my first microscope came when I was 6 years old. I was that kid. You all would believe that. You know why it came second? It's true. My parents got me the chemistry set first. They were very unhappy with what I did to the kitchen. That kid. You don't want that kid. I have a grandson who's going to be that kid. We're still impressed that the kindergarten teacher has not resigned after 4 weeks. And that ends the discourse on passive movement. We're now going to get into, aren't you happy, active transport. I see your enthusiasm is boundless. Aren't you happy after the game? On the top 10 of ugliest Steeler wins ever, it is in that. It has got a place reserved in the top 10. Ugly. It didn't stop me from enjoying it, however. So here, and the reason why you will see right away why I use these subsequent PowerPoints. Okay, this is now active movement. Active implies utilization of energy. So we will not get into vesicular transport today. Vesicular transport, anything that's enclosed in membrane, other than a cell membrane, is kind of a vesicle. A vesicle is a way we transport things in and out of the cells so that they are protected. The membrane protects them. So when you open up a membrane to either spit something out or you grab something, you envelop something in membrane and bring it in, which we'll just touch on and introduce, you have to expend energy. So even though it's not classic active transport, they are active processes requiring the expenditure of ATP. And so all of that, and that's because typically things we bring in and out of the cell are too big to get in normally. And typically they are not lipid soluble. And those don't really apply so much to concentration as to size, I would dare say. And you're going to see it applies to solids and liquids as we get on with the definition. But active transport, more than half our energy goes to one kind of active transport. And you will see half the ATP we make every day and spend every day goes to something called a sodium-potassium pump. And it's in every single cell. And it's constantly active in nerve and muscle cells. You can kind of see we're heading in this nerve and muscle cell direction. So when we use the term pump, it is, I heard that. When we use the term pump, it is synonymous with active transport. So if you see proton pumps, that's a form of active transport. Sodium, potassium pumps, form of active transport. We are very frugal in the expenditure of energy. Therefore, when we can transport something along with it, if you're opening up the doorway and putting in energy, you might as well carry something along as we bring something in, or kind of like the ocean hitting the beach. And you get the undertow, which is the water going back out and pulling sand with it. When something moves in, we can use sort of the backflow of it to push stuff out. But if you're forcing something in, you're going to have to create a certain other pressure within that cell to potentially push stuff out. So those are called antiporting, bringing something in and pushing something out along the same time, sometimes through the same opening, or SIM porting. Another term for SIM port that's a little easier, I think, to understand is called co-transport. It's being transported along with it. Like you're driving to school and somebody else needs a ride to school and you pick them up, you're co-transporting. And so against the concentration gradient is important to realize, and it requires energy. And we're going to see primary active transport is taking that ATP and typically opening up the passageway to bring something in. That is something that we see in those, what we would see, sodium, potassium pumps. That's really the best example of primary active transport. That's what it does. And as you will see, it's not just sodium moving in at the same time. It's not just sodium being spit back out. We're bringing potassium back in. You'll see that in a second. So it is a form of primary active transport. Yes, when doing so with other kinds of pumps, other kinds of active transport in the kidney, when we reclaim sugar, you're not supposed to excrete sugar in the urine. That means that indicates most likely you're a diabetic or trending toward one. And what we do in the kidney when we will actively transport sodium and co-transport glucose, effectively it gets back into the bloodstream. That's just a common example. So it's used a lot. We do it in digestive. We'll bring in sodium and we'll co-transport it in something else when you get to digestive. These are all AP2 types of things that are there. So they kind of are obtained indirectly. That would be a good way. And here, this energy causes a change in shape, and that change in shape causes solute ions. And in this case, there are two going in both directions. And this is the classic pump, sodium-potassium pump. It is really just an enzyme, and certainly is the most studied. Okay, and what you will see it is doing, as I show you the animation in just a second, is basically making sure that we have the appropriate balance in our cell. This is the whole crux of what's called resting membrane potential. That understanding that is the basis of understanding nerve and all. Every living member, whether it has the ability to change or not, will always be along the entire membrane, that cell. Positive on the outside, negative on the inside. It's just a difference. Doesn't mean it's dramatic. For instance, if you have more positive particles on this side of the membrane compared to the other side, you'll represent it as positive and negative. You're going to see it's about the particles that are there. Indeed, you will find out that inevitably we have more sodium on the outside of the cell and more potassium on the inside of the cell. But I'll let the animation tell the story. This is what occurs with what's called resting membrane potential. And because it changes so often, constantly, even just you're sitting there relatively just basically using your nervous system, thinking, and maybe a little bit for writing, you're certainly not being overly physically active. So I will move on, and hopefully the sound is appropriate. We'll buzz back to these in a jiff. Check the sound.
The human brain alone contains about 100 billion nerve cells, called neurons. A neuron, like every other cell, has positively and negatively charged ions, inside and outside. Further, a resting neuron has a greater negative charge on the inside surface of the plasma membrane and a greater positive charge on the outside surface. This partitioning of charge creates a voltage difference across the membrane known as the resting membrane potential, which can be measured using a voltmeter. On average, an intracellular electrode records a value of minus 70 millivolts. The resting membrane potential depends on two factors. First, it depends on the presence of sodium and potassium gradients across the plasma membrane. Specifically, there are more sodium ions outside the neuron than inside, and more potassium ions inside the neuron than outside. Second, the resting membrane potential depends on the differential permeability of the plasma membrane to sodium and potassium ions. Leak channels in the plasma membrane allow sodium and potassium ions to diffuse or leak down their concentration gradients. The membrane contains many more potassium leak channels than sodium leak channels. Thus, the membrane is much more permeable, or leaky, to potassium ions. As positively charged potassium ions leak out of the neuron, the inside surface of the membrane becomes negatively charged compared to the outside surface. If potassium was the only ion moving, the potential would stabilize at minus 90 millivolts. However, positively charged sodium ions leak into the neuron, which slightly offsets the negative charge and raises the voltmeter reading to minus 70 millivolts. Sodium-potassium pumps actively transport sodium ions out of the neuron and potassium ions back in,
They maintain
compensating for the sodium and potassium leaks. Thus, the pumps help to maintain the resting membrane potential.
A couple of things to note. So that's why there's always more sodium outside. There's always more sodium outside and potassium inside. Those pumps, even in cells, they work occasionally in most cells, in nerve and muscle cells, constantly, because they're constantly sending signals, all of them that are there. And what's interesting, why this is a two-way kind of a pump, we're getting, again, maximizing your amount of energy. If you look and you go back and listen, you can see there are, the red is taking 3 of those sodiums, and that little sort of burst of light is the energy, and pumping them out to maintain the sodium on the outside. In the meantime, they're bringing in 2 of the blues. Get back to where it starts. So 3 going out, and simultaneously that 2 of the blues, which are the potassiums, coming back in. And if you look at the way it looks resting wise, and we'll see that in the illustrations I kind of went through, and I'm going to go back to. This is a very good representation of how large a membrane is. Again, we're so used to looking 2-dimensionally. 3-dimensionally, this is massive. And the whole idea of why this occurs is about the potassium. It has to do with those leak channels, why potassium is very important. I've said it before. If you look at a series of blood tests, we have a wide range, something like, I forget, 125 to 145 milliequivalents of sodium. But potassium is like, 3.5 to 5.5. Forgetting what if you look on a typical blood test array. Doctors, nurses, clinicians, people who are familiar with this stuff, when they see somebody's potassium get too low or too high, or for that matter, the calcium, which has a very limited range, too low or too high, that is big trouble. Because it's going to alter inevitably resting membrane potential, and that's either going to make the heart more likely to be overly excitable and have arrhythmia, or be too slow, the nervous system to work too slowly or too rapidly. That's what you get excited about. There's no, we'll give them some potassium tablets and call me in the morning. No. You're going to start an IV and start putting potassium in it. I mean, I'm trying to give you a sense, because I spent a long time doing this professionally, to understand why these things are important. I had the misfortune of having to get potassium put into me in a big hurry when I was quite sick in an ICU. That was 20 years ago this coming summer. And you only go to ICU if you're near death. So I have a lot of interesting stories to tell you, having lived this. So here we go. Active transport. Sodium in, leaking out. The whole idea, they're following their concentration gradients. This pump is keeping them stabilized. This is the most studied one of all, and we've studied it particularly in association with genetic defects, which may or may not be associated with chronic fatigue. I mean, chronic fatigue is a real thing, and I don't know enough about it to teach anybody about it. So whether it's an autoimmune disorder, your body attacking the ability of these pumps to work. I don't know. And I haven't really studied the research about it. But understand, if you can't stabilize it, it's going to alter nerve and muscle function significantly. That's why it's so important. And here's that 3-2 thing. You can go through these, but you can see over here, the yellow is the sodium. Okay, I beg your pardon. The yellow is the potassium coming in, and in this illustration, the green is the sodium going out. So you have 3 spaces. Okay, eventually I'll get this right. The sodium is the yellow, and the green is the potassium. You have 3 spaces for the sodium, and only 2 spaces for the potassium. This 3-2 relationship of sodium and potassium, you're going to see a lot, and particularly when you start to look at really the first one of these very difficult things we look at, called the neuromuscular junction. It's the way that everybody learns about how a nerve transmits its chemical message and makes a muscle move. There's a whole lot of steps, and this is a really big player. So keep this in mind, and that 3-2 relationship is significant, very, very significant. And you can look it up that way if you want to think of it an easy way. I always thought of sodium and potassium over in the periodic table, very similar, except sodium has a lower number. That means it's smaller. So if you have the same space, you can move 3 sodiums, but you only can move 2 potassiums. And that relationship works both coming and going, either being pumped out and pumped in, or normally escaping in one direction or the other. And so that's the whole idea. You can look through all of those if you wish. It's fine. Secondary means we're using whatever primary active transport, not necessarily the pump. We're going to bring active transport to bring sodium in and carry something along with it, or push something out of it. The primary typically is moving the sodium. Anything else that comes with it is secondary active transport. So sodium can drag other molecules with it as it flows through the cell through carrier proteins, symporters, or what I like to call co-transporters, which is the term they used to use. And again, this is how glucose gets reclaimed. This is how you absorb amino acids in your gut. Okay? And so those are the secondary transports. Or we're kicking something out at the same time. That is antiport. So again, you'll get a look at those terms here. You can see it a little bit more expressed graphically, where you can see the sodium's coming in. Okay, here's a sodium-potassium pump. Here you can see that's creating a certain amount of energy, and at the same time, we're mobilizing a sodium pump with glucose. So I don't think it's a great example, but you kind of get the idea. This is primary. This is a secondary form. Not so much the sodium, but that glucose moving. Nice to see you. So I'll introduce this idea of vesicular transport. This is the other active transport. Opening and closing vesicles. It's constantly done. It's the way we alter the shape of the membrane. Membranes, again and again, but I use the same language to express it to you. Membranes are flexible. So we have things that you're going to learn about called microfilaments. They're tiny filaments made of a protein that assemble and disassemble. So that if they assemble, they push on the membrane and they create a change in its shape. And that can go around something and eventually engulf it and bring it in. And as the shape and we can do it the same way. It can basically envelop something when it gets bring it to the edge of the membrane, open it up and kick something out. Those are the processes of endocytosis and exocytosis. So that's the beauty of having a flexible membrane that can open and close and still retain its stability, not for the least of which is the presence, not the least important part of that is the presence of cholesterol. And that's what I'm trying to get a point across in a long-winded manner, is to understand the dynamic nature of living things. Changing shape, whether it's protein changing shape, whether it's cells changing shape, the movements we create. Shape changes are everywhere. And so and really to me, that's a critical point of understanding structure and function as they relate together. So it does involve energy. endocytosis, bringing it in. They all involve, to a certain extent, receptors. So I think the term receptor-mediated endocytosis maybe is incorrect in the fact that it's just as though this is something specialized. Probably not. The reason they are citing this term, as you'll see when we get to that specific example, is because of the hereditary basis of the receptors. When you hear about somebody who has what's called a hereditary lipidemia, they have a genetic factor where they always have an excessive amount of fats in their bloodstream, and they have to take certain medications for it. It has to do with those receptors. And I'll get to that example down the road. But by and large, it is driven by receptors as well. Endo, bringing it in. Phagocytosis means eating. Phage is eating. Penocytosis is drinking for liquids or oils. And receptor mediated, they all are, but I think it's very specific when we look at the way the construction of that. But we will not get to that today. And eventually, if we bring it in, why are we surrounding it with membrane? To protect it and to protect us. Things that we ingest, solid or liquid or otherwise, we have no idea if they're bad guys there with a party. I mean, you think because you cook food or anything like that, it can hurt you just the same. It could have heat-stable toxins that are embedded in the middle. There's a lot of that stuff going around. Okay? So understand those cooking guidelines are internal temperature of 165. There's a reason for that. But your cells, even when they bring in those tiny particles, each one of those particles is subject to scrutiny. Basically, whatever is brought in, it's going to fuse with a container with enzymes for digestion and defense called a lysosome, and it's going to process it. If it's good stuff to eat and nutritious, and we can use it for building blocks, it'll gobble it up, make it usable, and we'll spit out the waste material. If it's bad guys, hopefully it'll clobber them. Because bacteria and viruses are constantly in our presence. Every time you inhale, there are bacteria and viruses. Don't think something magic is because of that stuff. It's there. You're covered, 100% of your skin is covered in bacteria 100% of the time. So down the road, you'll learn about this, hopefully in micro. So we look at this. Exocytosis is spitting it out. A lot of things, we just use the cell as a passageway. We encase it in membrane as it comes in and release the membrane as it goes out. We call it transcytosis. And we'll see that in digestive, and we see that in urinary. Those are things that occur. And really, we have a whole way of controlling these traffic patterns, fancy name called vesicular trafficking. So these vesicles, okay, usually involve specific receptors. Most of the time they do. And the substance basically triggers that receptor to allow it to fold. And as you can see, there are pathogens that uses HIV. That virus uses a mechanism like that. It latches on to certain white blood cells in such a way that it joins with them because it has similar markers to receptors. And at that point, all of a sudden you got trouble. Enough of those markers get involved, that turns someone from being just exposed to the virus to actually getting the virus and all of the complications there too. Just to give you an idea of how significant it is, put this in a frame that you would understand. Diseases can get carried this way, and some more easily than others. And as you'll see, once it's pulled in, all those proteins get shed, and now we call it uncoating. At that point, a couple of things happen. Inevitably, it fuses with a lysosome. That lysosome, I said, could gobble it up if it's useful, could destroy it if it's bad. Sometimes we won't have a lysosome per se do anything to it, and that'll be transcytosis, where it kind of just goes through the cell as a conduit or passageway. Okay, but this is the way your white blood cells work. The biggest white blood cell we have is called a macrophage. Your immune system is constantly finding in little invaders and basically tagging them. They immobilize them, they neutralize them, they identify them and mark them. And these white blood cells are constantly on patrol, and they go along and say, I got one, gobble it up. That's what they do. We eat the destroyed material, we break it down, and we spit it out. The problem is when we can't get it spit out, or if that system of protection is overwhelmed, and you'll hear me say it again, which I talk about in micro all the time, it's a numbers game. Yeah, I mean, if you get exposed to a ton of those viruses, a ton of those bacteria, it's a thing called infecting dose. It's pretty significant. You'll learn about that in something called virulence factors in microbiology. Fascinating stuff. So, and you can go through the way they've labeled these things. That's fair, and see how it is. But basically, this is called something called a phagolysosome. We go over this multiple times. Certainly, a lot in AP2 and immune system. And that phagolysosome is really where you get enzymes, as you see here, for digestion, okay, or deliver its contents by transcytosis. I mean, so it's digestion or defense. And you're going to see it's not the only vesicle that plays a defensive role. So phagocytosis, eating, again, this ability to alter what they call false feet, we will show, and we'll talk about amoeboid motion. I will come back, probably starting here next time. I think I'm doing that with all the classes. And I will see everybody Wednesday. Thank you.